Debater of their choice. Each debater will then have 3 minutes to respond to the question. Once each of the panelists has asked one question, we'll move to the second round of questioning and conduct it in a similar manner. I just have one request of the panelists. If you could turn your name tags towards whoever you direct the question to so that they'll know, who's asking the question. That would be helpful. And so I guess we'll proceed in alphabetical order. And we'll start with Dr. Balnick. So, go ahead.
All right, this question will be directed at Dr. Nelson. So, Dr. Nelson, I very much appreciate your comments about intelligent design being very much in its infancy as an area of inquiry. In fact, I'd argue that it's perhaps even premature to call it infancy. We might say that it's a gleam in the eye of its conceivers. But that said, usually at that stage in the scientific idea, if we were to take your argument that we could consider this a scientific idea, one doesn't start a public debate. One goes to the laboratory. And I'm very curious why we're here. Because frankly, I do have better things to do with my time. I could be home working tonight. So that said, my question for you is that if you want to add design into the mix, you're more than welcome to. In fact, I'd go so far as to offer you use of my laboratory and PCR machine and micropipette and so on the condition that you tell me exactly what data you would collect to test your hypothesis, or what hypotheses you have.
Can I first speak to your comments about why are we here? A friend of mine, the Smithsonian, published a paper by a colleague of mine, Steve Meyer, in the Proceedings of the Biological Society of Washington. He underwent hell, personal hell, and is still undergoing personal hell. He's a first-rate scientist, first-rate evolutionary biologist, Rick Sternberg. Unfortunately, this issue has a real socio-cultural dimension where things need to be broken up. Hegemonies need to be broken up in part by public debate. I'm sorry about that, and I agree with you. It would be nice if we could conduct this strictly in the labs and in the journals. But the reality is that's not the situation that we face. Now, about what kind of data would one gather? What if it's true that minimal complexity is a genuine lower threshold? In other words, if you're going to have a cell at all, you've got to have, let's say, 300 proteins of very different types to function at all. That seems to me to be not a prediction that one would derive from any kind of bottom-up scenario for the origin of life, which I expect is the view you yourself hold. So if I were working in your lab, and you told me I've got a pathway, a natural pathway to a ribosome, or I've got a natural pathway to a protein, I would challenge you about that and say, if you have proteins at all in the cells that we actually know, not hypothetical cells 3.8 billion years ago, in the cells that we actually know, you need the following machinery. I can test that. Furthermore, you need this special machinery that we don't actually even understand in mycoplasma. Let's look at an unrelated bacterial group. If its orphans turn out to be essential, and look at a third group, and its orphans turn out to be essential, and those orphans are not congruent among those three groups. Under current theories of evolution, there would be no way of making any Darwinian sense of that picture. You have three fundamentally different biochemistries that have some parts in common and some parts that are separate, and you have a clear lower threshold that, if it is real, is irreconcilable with the bottom-up scenario for the origin of life. What is the natural pathway to RNA? It doesn't exist as far as we know. So my inference to design is based on what we actually know about the delicacy of that molecule. And it's you who are holding out hope for a natural mechanism for something that the evidence indicates will never arise from its constituents. So there's plenty of data to gather. That's not the problem. The problem is, what am I going to be allowed to do with that data, and what kind of inferences will I be allowed to draw? And the reason we're here tonight is this issue matters. And I'm sorry I took you away from your home, but here you are.
Question for Dr. Nelson. It seems to me that you really like analogies. And I'd like to particularly analogy of the minimum cell. So by analogy, following the same analogy, if we were to study, say, modern-day cars, there will be many pieces that are essential for the working of our car. And we could probably conclude that the minimum basic car would be about maybe a Honda Civic. So by that same model, if we went from the other tunnel, we would basically conclude that the Ford Model T shows an impossibility. So I think that analogies, are hard to follow through, and as Dan pointed out, I think the important thing is to do some lab work. What concerns me most is that it seems that the entire argument is an emotional appeal for our sense of wonderment and our innate ability to detect patterns. Right? And we clearly, you're pointing out that this protein clearly has a pattern. Why do we think it's not a sign? So, how are we going to pinpoint from a scientific point of view that? How can we distinguish that that's the sign and not a gorgeous snowflake or a buckyball that are phenomenally symmetric and show sign? Or by the same token, you're probably aware of the space on Mars. Is that the sign? Why should we not think that there is space of evidence out there doing all the design that you're proposing to?
Well, I'll rephrase what I take from your question. You can tell me if I've captured it or not. Humans are pattern-perceiving creatures, something that we do. In many cases, however, those patterns are factitious. On the back of the New Hampshire quarter is the old man of the mountain. It's not there anymore, it's sad. Right after they made the quarter, it fell down. It doesn't exist anymore. But you can see it on the quarter. Now, if you walk away from the angle where you see the old man, he disappears. That's a factitious pattern. But we know that there are real patterns that all of us, irrespective of our theological or philosophical predilections, are explained by reference to design. We do this regularly, and we can sift the patterns. When TWA Flight 800 blew up over Long Island Sound, in the New York Times the next day, and in the press for weeks, the leading hypothesis was a bomb. That the plane went down by someone's deliberate intent. When the FBI published its report 2 years later, that candidate hypothesis was gone, replaced by a natural mechanism. You don't let exposed wires go through a central fuel tank that's full of fumes because a natural regularity will kick in. What happened in that 2 years? They went looking for the evidence that would indicate intelligent causation, debris from an explosion, and so forth. They couldn't find it. So it is possible, in fact, we do so regularly, to rule out or to rule in design explanations. What makes it problematic in life, as Sahotra pointed out, is we don't have direct observational access to the relevant designer. But think about this. Get The Origin of Species, the first edition, and open the book to page 6. What Darwin says is, the view that I held as a young naturalist, and that I held right throughout the Beagle voyage, I now take to be erroneous. What did he do? He had a theory of design, a primitive theory of design of the time. He tested it against observation on the Galapagos, on the voyage, comparative anatomy, and so forth, and he falsified it. There are whole web pages dedicated to trying to falsify Mike Vickey's idea about irreducible complexity. In fact, I was at the AAAS in Washington in the fall and went to hear a talk by Chris Adami from Caltech, the whole context of which was, I wrote a paper for Nature with Richard Lenski to test Michael Behe's ideas about irreducible complexity. The fact is, these ideas are testable because it's going on all the time. In fact, you can get papers published in Nature or Science critiquing intelligent design. What you can't do is get a paper in advocating for it. Now that's the kind of fundamental asymmetry that should turn your radar on. How is it possible to criticize an idea in a scientific journal, as Adami did, and not to make a positive case for it? So these ideas are testable.